many people would want to go to Sunnyside? So getting a bus trip to Sunnyside on Friday afternoon. So I guess, okay, so we've got 2. Is it just 2? Just keep your hands up. 2, 3. So a trip to Sunnyside. Okay, it's about Mrs. White's house, yes. All right, so we've got about 5 or 6. All right. All right, well, look, thank you for coming back. We've got, I think, an interesting session this afternoon. As you know, we've got 2 talks. The first one is comparing human innate DNA. And following on from some of the questions that were a little earlier, I think Dr. Standish is going to be able to sort of open us up a little bit to whether or not, that comparison, what sort of level of similarity are we actually seeing. And so that'll be a little bit of a longer talk, and then that'll be followed by Dr. Brand, can the evolutionary mechanisms survive the new biological insights? Also will be fascinating. So we'll ask Tim up, but just a little bit of background to Tim. Tim, though he has somewhat of a confused accent.
It's not the only thing that's confused.
No, I've known Tim for a while, it's okay. Tim is one of the scientists at Geoscience Research Institute in Loma Linda and has a molecular biology background. I've heard him speak a number of times. And I know that we're going to get some excellent information. So thanks very much, Tim.
Well, thank you. I'm going to present you with a quotation. And I actually want some audience participation. I want, just when I read this, I want you to take a guess at who wrote it. So here we go. Anyone who observes the chimpanzee, the gorilla, or the orang would not find it difficult to believe that they have some common ancestry with the human race. Who wrote this, do you think? Oh, you guys are good. You're good. You've been studying your stuff. Yeah, this is Doris D.E. Robinson, actually, I believe it is, Doris Robinson. He was who? He was Ellen White's secretary. Okay? And I'm kind of fascinated by this because what it points out is that, sometimes we're not going to all agree on something like this. We can look at data and come to different conclusions, even though we share common beliefs in a fundamental way. Now, I personally do not find the evidence to be very compelling myself when it comes to similarities and between particularly chimpanzees, which are supposed to be our closest relatives, living relatives, particularly when we look at genomes. So let's take a look at the scientific evidence, and you can draw your own conclusions. So we're going to look at just some data that I believe is informative when it comes to this. I do want to start out with God's Word, though, because there's something very important that we need to get out of it. And this is Genesis 2:7. It's probably quite familiar to most of you. And the Lord God formed, and you'll see I'm giving you the Hebrew word there in yellow, for formed. The Lord God formed man of the dust of the ground and breathed into his nostrils the breath of life, and man became a living being. I have friends who have talked to me, and I have believed them over the years when they have said, look at this. Look at how unique the creation of man is. God is so intimately involved. And of course, my Catholic and also most of my evangelical Christian friends would believe that many of them would believe that when it talks about God putting the breath of life into the nostrils, that's like the soul going in. The soul is coming from God and going into the man. And man's creation is unique. It's special. This special kind of creation is what makes human beings, the princes of the universe, to quote Queen. Have you ever listened to that song? Man, that's really kind of like a great controversy song. It's amazing. Anyway, so look at this. Look at the process here. Man is formed of the dust of the ground, right? And then the breath of life goes into him. So man is like this entity where all of these things have come together. Now, for better or for worse, most people seem to have stopped reading the Bible about there. But if you continue on down, just 12 more verses to Genesis 2:19, it tells us some details about how animals were made. And it says, "And out of the ground the Lord God formed every beast of the field and every bird of the air and brought them to Adam." Oh, wow. So God formed them too. It wasn't just us. And then if we keep on reading in the Bible, we get to this. "And they went into the ark to Noah, 2 by 2, of all flesh in which is the breath of life." Now, what was that? Was that other people? Was there only Noah and a few other people on the ark, or were there other creatures there? Right. What were the creatures that went in 2 by 2? Yeah, the animals, right. It was the animals, they went in. Now, I do want to point out that a different word for breath is used here. It's a slightly different word. So you might argue, oh dear me, look at that, it's something different. So the special thing that the human beings got, that's what made us different. But in fact, if you look at this particular word, and I won't try to speak Hebrew to you because, well, I'm not Hebrew. But you can see that these words are used interchangeably. They are synonyms in a very real sense. So when you look at the Bible, it seems to be spelling out something very important about us and the rest of creation. Okay. Who are we made by? God, right? One being, one God, one designer, one God. And what are we made out of? The dust of the ground, and then we get the breath of life and we become living beings, right? So what about pigs? Who made pigs? God, right? And what did he make them out of? The dust of the ground. And do they have the breath of life in them? Yeah. Basically, we're looking at a situation here where the same God is making different organisms, but out of the same stuff. So it should not be overly surprising to discover that there are some similarities between you and me and pigs and chimpanzees and mice and bananas. Okay? Of course there are going to be similarities there. That is what, at least in my opinion, the biblical account leads us towards. Now, obviously it doesn't say, and therefore pigs and bacteria and gum trees will all use DNA or anything like that. But there's a certain expectation that I think a reasonable person would have. When you look at a painting by Leonardo da Vinci, you see similarities, and it's not surprising because they were both by the same artist using the, well, except when he went off and tried to use tempera paints on the walls of things. But when he was using oil paints, it was pretty impressive what he was doing. He kept experimenting with different materials, I guess. But you can see those similarities. It's not surprising that we find similarities between different organisms. In fact, it's probably something that we would expect. So just for those of you who've forgotten what high school was like, you'll recall that all living things are made out of cells, and the kind of cells that we're made out of, and pretty much everything you can see that's alive, has a nucleus in it. And in that nucleus there is DNA. And that's the same for everything. Now, there's this idea that if we look at the DNA, we know where that DNA came from. Okay? I know where my DNA came from. It came from my father and from my mother. Okay? And I know where their DNA came from because I actually met the people. Okay? My grandfather and my grandmother, who lived right over here on Alton Road. And of course, my paternal grandparents as well. We know where DNA comes from, and we also know that most people can tell the difference between me and my father. Even though ever so slightly less than 50% of my genome came from my father. That's the way it is with all of you. Our mothers just give us that little tiny bit more. But, oh, by the way, I forgot to mention something. If you look at the creation account, there is a kind of being in this room that is the one exception to the rule. Okay? Everything looks like it was created in the same way, all animals. But who wasn't created in the same way? Eve, right. The creation of women is unique in the creation. Yeah. It's a good thing. I'm desperately trying not to use the word ordination in the next sentence, but anyway. All right. So when we look at chimpanzees and human beings, yes, we can see some similarities between them. This particular chimpanzee here, if I recall correctly, is there at the zoo in Sydney. And that chimp-like organism on the right was a little boy named Tim Standish who looked very chimp-like in Penang. That was the most chimp-like picture I could find of myself. And we both have DNA in us. There are similarities between us, and so it wouldn't be surprising to find out that those morphological similarities were reflected or in some way correlated with DNA similarities, because we know that there's all this information that's stored in the DNA, and it has a mighty big impact on how we turn out. I blame my father for this nose, for example. Anyway, the argument that is made is this. Darwinists have claimed that the human and chimp DNA sequences are almost identical. Or only 1% difference separates these genomes. And therefore, chimpanzees and humans share a common ancestor from which we inherited our slightly different genomes. Just as I and my brothers share a common ancestor in my father, and we're a little bit different, all of us, from our father, and a little bit different from our mother, So, we're just looking at the same sort of thing. It's just that instead of going back a generation or two, we go back maybe a few thousand generations. They claim that we diverged about 6 million years ago. You see different numbers, and it turns out that when you look at these genomes, you actually wind up with different phylogenies based on different segments. And so some people have humans and chimpanzees separating for a while and then suddenly finding one another attractive again and making babies, and we're the hybrid results. In any case, I will argue that differences between the human and chimpanzee genomes are profound, and explaining them within the paradigm of common ancestry is problematic. Design by a single creator better explains both, and this is important, both similarities and differences between these genomes. Okay, because you can't just say, all right, I'm going to pick all the data that is consistent with my theory, and I'm going to ignore all the data that are not consistent with my theory. You've got to take into account everything. That makes it much harder sometimes, but sure, when you start looking at similarities between things, they are pretty well explained by common ancestry. It's not a bad explanation. But there are differences too, and you can't ignore those. So let's just quickly go through some vocabulary. You've probably heard the word genome, and it just means the genetic material, the DNA of an organism. DNA carries information about how, where, and when to make proteins. DNA is made out of chemical letters called nucleotides, and you've already heard, what was that about the Australian Capital Territory, something or other? Yeah. Well, I work at the GC. That's how I remember it. Anyway, the sequence of nucleotide letters encodes the information just as the sequence of letters in this sequence encodes information. So, just like we sequence things, letters together, and they can say good things or bad things. The same is true with DNA. Some people ask, why did I go for molecular biology stuff? Well, it's because I wasn't that good at English. Way too many letters in the alphabet. Anyway, so here's a statement, and I want you to just sort of consider it. This is from Scientific American. Scientific American is a popular science magazine. And there's an important lesson that I think we can learn from looking at this and perhaps some other things along the way here. So this is somebody named Catherine Pollard writing. She says, "A humbling truth emerged. Our DNA blueprints are nearly 99% identical to theirs." That's chimpanzees. I'm personally not humbled by it, but anyway. That is, of the 3 billion letters that make up the human genome, only 15 million of them, less than 1%, I'd call it about 0.5%, have changed in the 6 million years or so since the human and chimp lineages diverged. So that's stuff that we've already seen, right? The 6 million years and so on. Evolutionary theory holds that the vast majority of these changes had little or no effect on our biology. But somewhere among those roughly 15 million bases lay the difference that made us human. I can hear an organ playing. Anyway, so, just so you understand, I'm not making up that 1% difference thing. She's saying they're about 99% identical. And the 15 million comes from the fact that half the changes probably would have gone on in the chimpanzees, and half the changes would have gone on in the humans. Both have approximately 3 billion bases in their genomes. So 1% of 3 billion is 30 million divided by 2, because you've got two things diverging in two different directions. Therefore, you get 15 million. Also, I want you to note carefully the date. 2009 in Scientific American. let's look at another statement, but this is from the journal Science. Okay? Science is a serious journal for scientists. It's not written for the general public or teachers or stuff necessarily. There's actual real stuff reported there. It's not filtered as much, although it can be very filtered. So here we have this statement. For many years, the 1% difference served us well because it was underappreciated how similar we were to chimpanzees, says Paul Gano, a zoologist at UC San Diego. Now it is totally clear that it's more a hindrance for understanding than a help. And I completely agree with him. In fact, it's always been a hindrance. Using the metric percentage simply isn't the right thing. It's like saying Sven is 2 meters smarter than I am. It's just the wrong measure, right? Percentage just doesn't describe what's really going on with these sequences, at least in any meaningful way. So there's the 1% difference, and yeah, it's more of a hindrance. Now, what was the date of that? What year was that Scientific American article published? 2009. And look at this. This is 2 years beforehand. And in Science magazine, the journal Science, it's openly being stated this is bunkum, okay? But in the popular thing, even years later, you see this nonsense showing up. In fact, probably if you're a high school teacher, you'll see something similar to this in your textbooks. And it will never go away. I know because I'm writing a high school textbook right now, and man, you just can't convince these people who are editing your stuff that what they're trying to put in is rubbish. It's incredible. But anyway, there are actually lots of numbers that have been published out there in the actual peer-reviewed literature, and it would be interesting to go through where these numbers came from, but I won't do it. We can maybe talk about it a little bit more later on if you want to. But according to the peer-reviewed literature, human beings are 99.4% identical. Wow. Maybe, yeah, anyway. They might have sequenced my DNA, I don't know. 98.77% identical. Man, it just sounds so precise, doesn't it? 98% identical. That 98% number is used an awful lot. I remember seeing a bus going past where I work with things saying we're 98% identical or something with a picture of a chimpanzee and come and meet your relatives at the LA Zoo. Anyway, 96%. Yeah, isn't this remarkable? When the chimpanzee genome was finally published, all of a sudden the percentage of identicalness dropped massively. But even that has massive problems with it, terrific problems with it. 95% identical here, a 2002 paper. I should say that more recently I've seen it drop to 86% identical according to one paper. I mean, there are all kinds of numbers out there. Where are they coming from? Okay? They are coming from people who are comparing different things in different ways. That's the bottom line, okay? And they're using different kinds of statistical approaches, and it goes to show you can lie with statistics, I guess. Yeah. Anyway, here are a few facts, a few ways in which you can just have a few facts in the back of your head and start evaluating how meaningful these statements really are. The human genome is not 3 billion bases. It's about 2.9 billion bases long. The chimpanzee genome is larger. It's about 3.1 billion bases long. And you get different numbers from different sources. However, again, there are good reasons why it's hard to nail it down. But size estimates vary from one source to another. But the chimpanzee genome is consistently estimated to be 6 or 7% larger. So how do you get 99% identical when it's 6 or 7% larger? Obviously, you are not actually comparing letter to letter or nucleotide to nucleotide. The human genome is divided into 23 chromosomes. You're probably familiar with that. You've got 20, most of the cells in your body will have 46 total. Chimpanzee genome is 24 chromosomes, so they have an additional chromosome. In fact, the other great apes all have this, one more chromosome than we do. Now, there is an explanation for that, right? And I want you to consider the explanation. We actually call in chimpanzees these two chromosomes that kind of correspond with one of the human chromosomes. We call it PAN2A and PAN2B there. And the theory is that our ancestor, our common ancestor with the chimpanzee, had these two chromosomes, and that somehow or other they merged together to form the human chromosome number 2. Interestingly enough, in the course of this, no genes were lost, and about 150,000 base pairs were added along with some genes from chromosome 9. So this is an example of one of those theories to try to explain a situation that sounds kind of elegant at first, but then when you start looking at details, it rapidly becomes a more complicated theory. I should tell you, there are people out there who are saying, listen, we're supposed to be seeing signs of this fusion in there and we're not seeing them. Other people are saying, yes, we are. Look at them. They're plain as day. So I don't know who's right. But what I do know is, irrespective of whether there was a fusion, this is not evidence of common ancestry between humans and chimpanzees. And the reason for that is quite simple. The fusion event had to, if there were two chromosomes that fused, it had to be in the human line, right? So this doesn't tie us back logically to a missing link kind of ancestor with chimpanzees. Obviously, either of these models that I've just shown you, this one, in which, let's say, Noah was so traumatized by the flood that two of his chromosomes fused, or or this model in which the common ancestor had it, and then we split from the common ancestor, and then there was a fusion event. They're as far as the data do not differentiate between these two possibilities. Here are a few more facts. 3 human and chimpanzee chromosomes, numbers 4, 9, and 12, appear to have been remodeled. And this is where, if you want to read some science comedy, it's well worth reading the paper when they published the high-resolution sequence of the Y chromosome, chimpanzee Y chromosome. You can almost hear scientists' jaws, bouncing on the floor. It turns out that the Y chromosome is profoundly different. So sorry, ladies, you don't have one, but I do. And it's just a very interesting thing. The ends, and this is really important, the ends of chromosomes, they're called telomeres. It turns out that humans have shorter telomeres than chimpanzees have on the ends of their chromosomes, which is kind of an interesting thing. There are lots of kind of interesting implications of that, but this is one of the reasons why their genomes are a bit larger than ours, one of several. Then just inside the ends, just inside these telomeres, there are subtelomeric sequences that are also different in chimpanzee chromosomes than they are from human chromosomes. And probably one of the most surprising things to me, and I think a compelling piece of evidence that maybe we don't share common ancestry, is the fact that recombination hotspots are different. You probably remember being bored in biology class while your teacher talked about the difference between meiosis and mitosis. But you're probably thrilled by that little bit in meiosis where all of a sudden the chromosomes got shuffled, that crossing over process. I hope you found it interesting. It turns out that that's not actually a completely random process. There are certain places where you actually have the chromosomes breaking and being recombined, right? And in chimpanzees, those happen to be very different than they are in human beings. Which is an indicator of quite a different architecture to the genome. Even though some people have advocated for this, it makes formation of hybrids between humans and chimpanzees a bit unlikely. I mean, you never say that would never happen, but it seems like a stretch. All right, here are some more numbers. The genes in the human genome are about 88% the same as rats. I'm just wanting to show you how you can just give these percentages and they are, you can make almost any argument based on them. About 60% the same as chickens. I know some people who I think are at least 62%. About 80% the same as sea squirts. These little marine organisms, they're not even vertebrates. And if you want to get sort of all kind of whatever about it, only 29% of human proteins are actually identical, possibly less, to chimpanzee proteins. What does all of this mean? Okay? What it means is there might be one amino acid that's different, and so you can call it different. I don't know, I don't think that's a particularly strong argument, but I just want to point out you can slice and dice your data in many different ways and make any number of different arguments about them. I would expect that most chimpanzee proteins would work perfectly well in your body. So anyway, I've already said the similarities are well explained by common ancestry and well explained by some kind of common designer. At least that seems that way to me. But what matters when it comes to differences in DNA sequences is not the percentage of difference. What matters is the same thing that matters with any other sequence that encodes information. How different the information is, not how different the sequence is, right? So, let me give you an example. First time I went to America, I went with my uncle down to the little tiny point that Virginia comes down into as you head west to a place called Big Stone Gap. And I was there with my aunt and uncle, and I had multiple people ask me if I loved my aunt. Didn't know I had an aunt. Nothing against ants. But really, it was very confusing. And eventually, I did figure out that, I didn't have to respond that I love my aunts. What they were asking about was my aunts. And I do love my aunts. This is my Auntie Loma. If you ever see her over there in the Charles Harrison, on the other side of the road, tell her I love her. Lovely aunt. And I do that. But what's the difference between, these are profoundly different meanings, right? Profoundly different meanings. And when it comes to DNA, we see that these differences are becoming more and more profound. But let's go back to this Gano guy, who I gave you the quotation from earlier. Researchers are finding that on top of the 1% distinction, chunks of missing DNA, extra genes, altered connections in gene networks, and the very structure of chromosomes confound any quantification of humanness versus chimpness. There isn't one single way to express the genetic distance between two complicated living organisms. percentage just doesn't do it. It simply doesn't do it. So let's talk a little bit about profound differences. Let's remind ourselves about the difference between percent and absolute numbers for a start. Small percentage differences represent very large numerical differences. So if the human and chimp genomes are approximately 99% identical, which they are not, that means they are 1% difference. These genomes are about 3 billion nucleotides long. 1% of 3 billion is 30 million. We've already talked about this. So, wow, 30 million, that's a lot of differences, right? In fact, the bad news is this. If we were to get the number of differences that we actually observe between humans and chimpanzees, it looks like our women would need to be giving birth to somewhere between 100 and 200 babies each, just to make it mathematically possible to get this kind of difference. That would be with essentially 100% selection against all other variants, just to get the number of fixed differences that we observe in the genome. I could, but I've only got about 5 minutes. But the bottom line is that when you have a whole bunch of people who are reproducing, you've got to generate those 30 million differences, right? There's not enough time unless our ladies are willing to crank out that number of babies, and there is perfect selection going on. So you've got to be willing to slaughter 99% of the babies and keep 1%, basically. Thank you, ladies. Anyway, yeah. If half the genome difference arose in humans and half in chimpanzees, that's where we get the 15 million differences that we talked about earlier. Now, I want you to note that this is, in fact, an oversimplification. There's some subtleties in all of this, but it does accurately illustrate the nature of the problem. Okay, so at least that much. These differences must have arisen from a Darwinist perspective over about 6 million years. It's just not going to work. So for every percentage you want to say we're different from chimpanzees, you're talking about another 15 million differences in the human genome, and another 15 million in the chimpanzee genome. These numbers get big fast. Let's talk about orphan genes. And I think somebody else is going to mention these, so I won't spend too much time on them. But anyway, some changes in genomes might be relatively easy. For example, I ate a dog to a dog I ate. They mean the same thing, and it's really only one change. I just made one break and reconnection there. This is equivalent to the relatively easy DNA change from AGG to AGA, in which both code for the same amino acid, which is arginine. Other changes appear to be much more difficult. So, for example, going from absolutely nothing to this Bible text, call on me and I will answer you and show you great and mighty things which you do not know, that's a big change. Okay? Getting that from absolutely nothing is really hard. Orphan genes are genes that are unique to specific organisms. Thus, they are not inherited from ancestors shared with other organisms. Common ancestry doesn't explain that. The human genome appears to have a significant number of orphan genes, probably in the hundreds. Right? So this is one orphan gene from human beings. This is the sequence, and if you could just memorize it, we'll have a quiz at the end. This is just what one of them looks like. That's the sequence of A, T's, G's, and C's. Obviously, something like this is a pretty big thing to get from nothing. And there are probably hundreds in the human genome and in the genomes of pretty much every other organism that we've looked at so far. So here are some conclusions. The human and chimp genomes are similar to each other, more similar to each other than to other organisms. It's not surprising. We look more like a chimpanzee than a banana. So there you go. Similarities between human and chimp genomes can be expressed in many different ways, but expressing the meaning of the similarities and differences is a more challenging task. Small percentage differences add up to large numbers of individual differences. Common ancestry, common design, and common constraints all explain similarities, okay? And I didn't unpack the common constraints thing, but anyway. Differences are not well explained by common ancestry. And this is one reason why I'm pretty comfortable with texts like this in the Bible, which I'll end with, for by him all things were created that are in heaven and that are on earth, visible and invisible. I'm guessing that would include DNA. Whether thrones or dominions or principalities or powers, all things were created through him and for him. And I thank God for doing that for us. Thank you.
Well, thanks very much, Tim, and hold your questions. We'll have time to ask Tim some questions shortly. But our next speaker is Professor Leonard Brand, who is a Professor of Biology and Paleontology at Loma Linda University, and we look forward to his presentation.
Well, hello, and I'm enjoying being here, speaking to this group. Can evolution survive the new biological insights? Sounds like kind of a, well, like I'm saying, well, maybe it can't. Well, let's see how it's going to go here. The goals of the talk: evaluate the current state of the evidence for the evolution of life forms by random mutation and natural selection, and understand the newest evidence causing big trouble for Darwin's theory. Now, for ages there have been arguments between evolutionists and creationists. That's not really what I'm talking about today. There's a growing conflict between, say, traditional evolutionary biologists and a group of scientists who are not creationists, but they understand some things that we're going to talk about here today. Put this in context. Look at just a little bit of history. Darwin worked in the mid-1800s. Molecular biology was unknown. Genetics was still decades in the future. They knew really nothing about what makes life. Now, the modern synthesis, or the neo-Darwinian synthesis, was developed in the 1930s and 40s, combining these fields: population biology, genetics, mathematical biology, and paleontology. Molecular biology was still in the future. And this is pretty much the evolution theory as it is accepted now. And molecular biology especially has developed since the 1950s with rapid advance recently. So that puts us in a little bit of historical context. What do we know now that we didn't know a few decades ago? About maybe 15 years ago, I gave a talk to a group of teachers, school teachers, Adventist school teachers, and somebody asked the question, has there been new evidence discovered that supports evolution more? In other words, why is it, they were asking, that evolutionists seem to have more commitment to the theory now than they used to? Well, recent advances in the last few years enabled me to give a better answer than I could at that time, especially spectacular advance in genetics and molecular biology. Just to summarize again a few definitions. Microevolution, adaptation, changes within a species. These are two lizards. They are the same species. This one lives on the gray granite rock where I live. This one is on the red rock in Arizona. They have obviously adapted, even though they are the same species. Macroevolution, the evolution of new major groups of organisms, orders, classes, and phyla. And when creationists complain about evolution, we're really talking about this. There's lots of evidence for microevolution. And what's the real difference between these? Well, microevolution does not require evolving any new organs, any new physiological systems, any new gene complexes, whereas here you have to evolve all those things. So that's really the difference between them. Okay, so microevolution, I believe, is compatible with creation. Adaptations since creation, and there's been an awful lot of that. Macroevolution, I will argue, is contrary to biblical creation. So how does microevolution work? And we can expand that beyond microevolution. The processes are this in Darwin's theory: random mutations and natural selection. Okay, so random mutations and natural selection, those are the core of neo-Darwinism. So in Darwinian theory, the reason for this is the process must be random. If changes happen because they would be beneficial to the organism, then somebody's monkeying with the system. There's some intelligent involvement there. The changes must be random in the sense that they do not know what would be beneficial to the organism. There cannot be genetic foresight in Darwinian theory. And only natural selection, a naturalistic process, determines which random change will succeed, according to Darwin's theory. Foresight would imply creation or intelligent design. Now, I made this little analogy of evolution, and it's just like something from a car factory. And so there's a machine that makes pistons, and it has a computer that controls it, and maybe you have lightning strike, and it changes one of the instructions. And so you end up having wider pistons. This would be an illustration. I need to pick something up from over here. Okay, that is basically that's a very simple analogy for how evolution, according to Darwin, would occur. There are problems for Darwinian theory of random mutation and natural selection. They are rapidly increasing, especially from advances in molecular biology. So how would random changes produce this feather, for instance? Something new, a new feature that did not exist before. This is incredibly beautiful. It's very stiff and strong, and yet very light, highly efficient. That's one of the big issues that we'll have to deal with here in our talk. So what are these problems that are developing? And you've other talks here before me have set a good stage for this. What is junk DNA? And for years we've heard about junk DNA. And you'll see this, you've seen this number. 98% of human DNA was called silent DNA because it does not produce protein. And that's been interpreted as junk DNA, functionless remnants of evolution. So this being true, are you willing to have 98% of your DNA removed? Probably not. And this junk DNA is needed by evolution as a resource for making new genes. Well, there have been problems developing in this concept. For instance, the percent of junk DNA is proportional to organism complexity. So here we have the percent of DNA not coding for protein. And so this was interpreted as junk DNA. Okay, you see very little prokaryotes, more in one-celled organisms, more in fungi, etc., until humans reach this pinnacle of 98% junk DNA. Now, why this graph? It seems to imply that the junk DNA is somehow involved in producing that complexity. And this was published before even the newer information tackling junk DNA was published. But through the years, there have been interesting papers that I've been following. Challenges to junk DNA. I like this one. It's the junk that makes us human. Could it be the non-coding DNA that makes us human? And papers like this come out periodically until finally, in 2012, the results of the massive ENCODE project were published. And conclusions drawn from this are that all or most junk DNA is regulatory DNA, controlling the protein coding DNA. So, at least to some extent, the supposed junk DNA controls whether our protein coding DNA will make us a human, a chimpanzee, or a mouse. So this is regulatory DNA that's controlling other DNA. So junk DNA kind of has faded out largely. And this is a problem in one sense because the evolution process no longer has that pool, it appears, of junk DNA from which to mutate and make new genes. So let's look at problem number 2. The tree of life is a very important part of Darwin's theory. All life came about through changes from the original simple organisms, so we're all related by this tree of life. But evidence for the tree of life, common ancestry, is facing some serious setbacks. So Darwinian understanding of macroevolution requires that all new features arise by a long series of gradual changes. Darwin talked about this. If any feature is found that could not be explained this way, this would refute his theory. There must be this long gradual series of small changes. The tree of life is claimed then to be a record of these changes. You've already heard about orphan genes, genes that seem to just appear with no evolutionary ancestry. So every gene, according to Darwinism, every gene should be able to be traced back. The evolution of that gene should be able to be traced. At least 10 to 20% of genes are orphan genes, hundreds of those in humans. Some are very important. I've seen papers claiming that there's a responsibility for the large human brain. I doubt if it's just one gene, but that's what this paper said. So here, here you have in these various organisms genes which cannot be traced to an ancestor. One example are some genes in honeybees. These genes give the honeybee the ability, among other things, to make honey. And they just appear. And so where did they come from? That you can't find those in other insects. It just appears that it seems that they just suddenly appear. It looks like maybe somebody put them there. Well, maybe that's not too bad an explanation. And so orphan genes are a serious problem for evolution. I've seen some papers by evolutionary biologists. One of them was looking at working out the evolutionary divergence patterns in some organisms. And they said in this paper that they use only genes that they can find in relatives of these organisms. Okay, what does that mean? They're ignoring the orphan genes. They're only using those genes that you can find in relatives. Okay, what that means is that they're throwing out a big percentage of their data, using the data that fits what they're trying to find out, and ignoring genes that are a deadly challenge to evolution theory. Okay, another problem is epigenetics. You've heard about epigenetics. I'm glad we had that introduction. A management system that determines how to use the DNA. Now we've heard that in humans, apparently most of these changes are negative. In other animals, there have been epigenetic changes studied that are positive, very beneficial. So sensors in the cell detect environmental factors, and they place these chemical tags on the DNA. In this diagram, there are these little pink tags. I don't think you can see it from there, but those would be the markers, methylation or other processes that turn genes on and off without changing the information in the DNA. And you've all seen this text, punishing the children for the sins of the father to the third and fourth generation. I wondered for years what that meant. and as you've heard before today, we know now what that means. This is a paper published in this prestigious journal Nature with this illustration, The Sins of the Father. Okay, can you guess where they got that title? This describes a very interesting experiment. Mice were exposed to a very gentle odor, the odor of peach blossoms. And every time they're exposed to this odor, several times a day, they get a mild shock on their foot. They only did this for about 3 days. And after that, when these mice smelled that odor, you get a very negative reaction. Then they mated these mice to female mice, who had never smelled this odor, and the offspring were not given any shocks or anything. But when they smelled this odor, they would respond the same way that their fathers did. Okay, so there's no mutations involved here. This is epigenetic changes. And also there were changes in the brain, in the olfactory part of the brain. So this was a significant change. And that lasted for several generations. So these epigenetic changes can last indeed for several generations. How many? We're not sure. Another example, blind cave fish. How did they become blind? Now, I used to teach my students the standard explanation, and that is that fish living in a cave, there's no light, and so if they have a mutation that damages their eyes, there's no reason why selection would eliminate that mutation. So that's how they became blind. All right. I also tell my students sometimes that half of what we're teaching you is wrong. The only problem is we don't know which half until science. The eye genes are intact. There have been no mutations, but the eyes have been turned off by epigenetic processes. No changes in the DNA. And there are many examples like this in animals that have been found. Okay, why am I saying that this is a problem for Darwinism? Because, at least in some animals, these changes, some of them are beneficial. And they are inheritable, at least for a while. And they're non-random. Okay, so environmental influences that cause beneficial mutations, now that is anathema to Darwinism, because if this environment is stimulating beneficial changes, somebody involved in making this system at least understood how to make a system that could do this. And changes that are beneficial, changes that happen because they're beneficial or closely associated with that, that's a serious problem for Darwin's theory. How can this happen unless there is somehow intelligent awareness behind the design or operation of this system? So epigenetics then is causing problems for Darwinism, and we're going to take a look at this again a little bit later. Another one, this is certainly not new information, but irreducible complexity. This structure is an amazing thing. It's actually an electric motor, a proton motor to be more exact, I understand. But it has all the features of an electric motor, and it spins this thing around and does important things for bacteria. Okay, that's an argument used by Behe. Others have criticized him and talked about differences in this fiber and lots of things. But I think they're missing the point. No matter how much variation there is here, you have this electric motor, which is, it can't develop one piece at a time. It all has to be there in order to work. And we can, of course, go to a host of other illustrations of this process, things that have to be there pretty much complete in order to work. Okay, how about this beautiful little critter, chipmunk? Now, at least some significant parts of this chipmunk have to be there before the chipmunk will work. So this is irreducible complexity is pretty hard for Darwinism to explain. And when I look at arguments against it, I find that they're dealing with peripheral issues. They're missing the point many times. They're really they really don't have an argument against this concept of irreducible complexity. It's hard to, they have a problem with it. Darwinists cannot really accept irreducible complexity, and yet they don't have valid arguments against it. Okay, number 5. This is kind of pulling together some things and talking about this group of scientists that I mentioned. A group of what they're being called new evolutionists. So these are, I'm not talking about creationists. There are creationists who think this way, but I'm not talking about them. These are scientists, not creationists, who recognize that Darwinian random mutations and natural selection don't work. Now, natural selection obviously is a functioning process, but if you have to start with random mutations, it doesn't work. New genetic information must arise some other way. Here's some things from a book by James Shapiro, Evolution: A View from the 21st Century. I find this a refreshing book. He's an eminent molecular biologist. He's also very frank and says what he has found from his research. The modern evolutionary synthesis included an ad hoc assumption about the random nature of hereditary variation. So ad hoc, meaning it didn't come from data, it came from philosophy. Your theory needs it, but the data it did not come from data. And he says this statement, which I certainly could say as well: it requires great faith to believe that a process of random accidental genome change could serve that function, adaptation. And he says hereditary change results from active cell processes rather than a series of random accidents. So he's just rejecting the idea that random change can do anything of any significance. And he uses this term natural genetic engineering for what's happening inside the cell. So cells are now reasonably seen to operate teleologically. Okay, that means the cell operates with purpose. Their goals are survival, growth, and reproduction. And he says the cell decides how to use the DNA. That's a remarkable statement. And he's a very prominent researcher in this area. So how do they explain the evolution process? Well, some of this is just a summary. Active cell processes, not random accidents. Natural engineering, cells have goals, and they work towards those goals. They decide how to interpret the DNA. Random mutations, he feels, are clearly not the source of genetic change, except some very minor things. Okay, all of that, I feel, requires a radical change in this analogy. Instead of this simple making meaningful changes by a random change, which would be, I would suggest, kind of like if I wanted to see if I can improve this computer. So I jab a screwdriver in it to see if I can make it work better. I don't know who owns this computer, but they might object. So really, I propose that the genetic process in a cell is more like this. Yeah, you have a master computer, but there are sensors. They're finding that organisms sense the environment, and these initiate changes. So the information they find is sent to control modules, and there are probably something you could call stage 2 controllers, stage 3 controllers. And then all this is processed in complex ways and sent back to the master controller. And they're finding there are massively more error correction repair mechanisms than they'd reasonably formerly understood. Incredibly good error correction mechanisms. And there also is what I would call standby piston specifications, software. Standby information. Okay, so what does that mean? Organisms have standby genetic information which is potential for change. And I'll show you my favorite example of that. Dogs. Dogs are believed to have come from wolves. Okay, were there dogs on the ark? Well, I don't know, but it really doesn't matter much because some of the dogs are so similar to wolves that it's a kind of a moot point. but apparently they came from wolves. Now, would wolves be happy with some of their ancestors? Well, I think they'd be kind of embarrassed in some cases. But it does seem that they've come from wolves. Now, how does that work? There are some mutations involved, but mostly it's believed to be epigenetic. So how do you get all this variation from these? And there are hundreds of varieties of dogs, many of which have come by human breeding in the last couple of centuries. Okay, how can evolution happen that fast? Well, it cannot. It cannot happen that fast. These are all changes that, based on information that had to have been there from the beginning, before humans wanted to produce all these varieties of dogs. The potential had to be there already. This is not evolution. It's something very different. And I don't know of any other animal that has nearly this much potential for change. I read one article by an evolutionary biologist who was puzzling over this. How did dogs get so much genetic variability? Let me suggest to you that God made wolves with dogs in mind. He knew we would need companionship and protection and many other things that dogs do so well. And so how else would these wolves get all that information in there? All that potential, what I would call standby genetic information. It had to have been, it could not have evolved. How could you evolve all this information unless you are making the animals at the same time? So this had to have been there, and I suggest to you one idea that might seem radical, but I think the only evolution that happens is evolution based on the potential that was put there in each organism from the beginning. And dogs are special in this respect. They have tremendous variability. Cats have much less. Sure, you got lions and things, but if you look at the smaller cats, there's a lot of different varieties, but you shave their hair off, and they're all basically the same, pretty much. And so we're looking at, there are some odd ones that have some mutations. But basically, there are no Doberman cats or bulldog cats. You just don't have that kind of variability in cats. So why did God make cats? Well, I'd suggest he made them to keep us humble, to remind us that we're not in charge. Okay, so genetic variability that may perhaps, if I'm right, was there in the beginning as standby information. So if random changes do happen, and they certainly do, there's a lot of random mutations, which is basically random damage. If they do occur, what's the most likely result? Well, they're listed here at the bottom. System failure, you die. Or reduced efficiency, you don't function as well as you used to. I've noticed some of that. The third one, which you can't see, is the mistakes are filtered out by the error correction mechanism. So this, again, doesn't leave a lot of room for major changes by random mutations. Okay, I'm going to use this kind of diagram to illustrate something further about all of this. Let me just explain the diagram. Here we've got microevolution and macroevolution represented here. And over here we have explanations of things. And there are two green lines. In my diagram, if this theory moves across this green line, you've moved away from naturalistic explanations down into something that is dealing with creation. Either direction. If you move your diagram up into here or in here, now you've gone away from naturalistic explanations. Okay, and for years, many scientists have felt that macroevolution is just the accumulation of microevolution over a long period of time. Accumulation of microevolution. Okay, so they're going along pretty much the same. More recently, the evidence has been pushing those two apart. Microevolution is now, by many people, believed to be a different process. When I say people, I'm talking about evolutionary biologists. It's not just accumulated microevolution over time. And further evidence is pushing it farther apart. Bordering on this area where you're moving out of strictly naturalistic explanations, getting dangerously close to something that might look like it involves creation. And actually, the evidence in microevolution is moving to the point where it's more compatible with creationist explanations. Evidence for rapid microevolution. I've seen a paper by evolutionary biologists pointing out that we used to think this took long periods of time. Now we know that it can happen within the period of one research grant. An example of that is a study on Darwin finches, beaks in the Galapagos. And indeed, changes in weather patterns and changes in food preferences, they could document changes in the bills over a fairly short time. Epigenetics is involved here, experiments that show that this can happen rapidly. Microevolution within created limits, that's my suggestion. One of our graduate students was studying variation in rattlesnake venom. And he could see evidence of pretty rapid changes. These rattlesnakes over maybe 100 square miles or more, maybe a couple hundred square kilometers, you find areas where they're quite different. And it seemed there were implications here that this is something that happened pretty rapidly. Now, he was corresponding with an evolutionary biologist, actually in Australia, who didn't see it that way, and he said, oh, these dumb creationists, they think this must have happened over thousands of years during the Ice Age. But actually, that person didn't understand what's being said by other evolutionists. This can happen rapidly. It's now being recognized. And so I'd suggest this is more, at least as compatible, or maybe more compatible, with creationist explanations. Now, we could argue about that. That's not, I wouldn't make a strong point of that. But when we look at macroevolution, it's more significant. Because of intelligent design, evidence for intelligent design, no junk DNA, irreducible complexity, orphan genes, epigenetics, all these things, internal developmental constraints, which we don't have time to go into. Macroevolution theory is collapsing, and I'm not the only one who says that. The article, the book that I mentioned by James Shapiro says exactly this. It doesn't work. Darwinian theory does not work. It has to happen some other way. How does it work? He just says it's a mystery. That's the way he deals with it. I would say that this theory is indeed collapsing. The evidence now favors an origin by creation. Okay, I wondered how evolutionary scientists are responding to these challenges. No junk DNA, orphan genes, epigenetics, the failure of Darwinism. And so I got latest edition of all these evolutionary textbooks, and I looked through them for terms, for concepts, if they were being expressed there or not. None of these books mentioned orphan genes. Okay, orphan genes, that's a deadly challenge to evolution. How could it be that the evolution textbooks don't mention that term even? That's amazing to me. Epigenetics, some of them talk about it, but it's always minimized or ignored. However, there are 4 other books here which are from a different group of evolutionary biologists. These authors here of these books, they recognize that things like epigenetics are real and they're here to stay. And they're trying to develop a new synthesis of evolution based on epigenetics. Now, about the year 2000, I was at a meeting of the Society of Vertebrate Paleontologists, and they care not only about fossils, but about how changes occur. And Robert Carroll, a very one of the leading vertebrate paleontologists. He gave a talk on evaluating the modern synthesis, the modern Darwinian synthesis. And he made a statement that I found very interesting. He said, that synthesis needs to be redone. And he said, this time we're not going to blow it. Not going to blow it. What did he mean? I wondered. And now I think I understand what he's saying. These people are recognizing, yeah, we need to redo that synthesis, and this time we don't want to blow it. And so they're kind of trying to come up with a new synthesis. Now, these people over here don't like these authors any more than they like creationists. There's a developing conflict within science from these two very different groups of people. And coming back to my feather, one problem that both groups have is they don't know how to explain how you get new features, novelties, new things like a feather. How do you get a feather? How do you invent bones or kidneys or livers? These are all new things that are tremendously complex. And how do you do that? There was a conference, a big conference between these two groups about 2 years ago in Europe. And they faced off and they tried to work out their differences. And I wasn't there, but people who were there say that this group would present their evidence and their challenges. This group really never responded to those except to say, well, natural selection will take care of that. Well, how is natural selection going to make a feather? Natural selection does not make new things. Natural selection does nothing whatsoever to make something new. What is selection? Natural selection eliminates things that don't work well. It does absolutely nothing to make something new. That has to come in evolution from random mutations, okay? And you can believe if you want to that random mutations can make this. I have problems with that. And they argue between themselves, and neither one of them really has an answer for that basic problem. So macroevolution is facing increasingly serious challenges. Eminent evolutionists, primarily molecular biologists, are raising those challenges and rejecting Darwinian theory. So why do evolutionary biologists seem to be more committed to the theory than ever? Remember that question that some teachers asked me. Is it because of new evidence? Why are they so committed to their theory? It's because of philosophy, not evidence. The evidence is going the other way. It is not giving them more support. Their commitment to their theory is a matter of philosophy. How could they accept the demise of macroevolution theory? How could they accept the demise of Darwinism? They cannot. Because if they do, what's their option? Well, they don't like the new extended synthesis, and that doesn't really work very well either. So they're pushed to creationism, and that, of course, is not acceptable. So I diagram that this way. These processes seem to be going at least in a direction where it can fit creation, maybe even is only explained by creation. And these are some people who are indeed challenging standard evolutionary biology, Darwinism. Shapiro, Denton, Mueller, Pigliucci, Noble, Tour, they recognize that Darwinism doesn't work, but they're not willing to step across this line, and so they're still struggling. How do you explain it? And here I would put what I call hardline Darwinists. Okay, they're not willing to move away from their Darwinian theory. And so they maintain their commitment from philosophy, not from evidence. So this is this conflict that I see developing in science. And I gave a talk to another group a while back suggesting that this is an in-process potential scientific revolution, like what Kuhn talked about. And so who knows how it's going to happen. I don't see that these people are ever going to give up their theory. And what are these people going to do? I don't know. But there is this definite conflict and struggle going on within science, not necessarily just between creationists and evolutionists. Okay, so conclusions. Darwinian theory of evolution through random mutations and natural selection is facing deadly challenges. There's a growing conflict within science between hardline Darwinists and the new evolutionists who recognize the collapse of Darwinism. Creationists have increasing reasons to be confident of the literal creation of life forms. This is a new day for us. Okay, how can we grasp the full truth about origins? How can we really understand what's going on? I mean, we can argue about specific little issues here and there, but really to understand the whole thing, it requires getting a broad-scale knowledge of the processes of life. All this tremendous series of biochemical reactions. You saw some diagrams this morning of those, but those are they just gave you a sampling. That's just the beginning. The biochemical things that go on in a cell, for us to be alive, have awesome complexity. They are just amazing. And it has to work for us to be alive. So we have to understand that. The whole complex system of physiology and anatomy, all these things, how they work together in detail. The more we understand that, the more I suggest that large-scale macroevolution and chemical evolution will sink into an abyss of impossibility, right next to a plan to build a railroad to Mars. It's not going to work. And I would suggest that neither one of those is going to work. So, further conclusions, and I think this is a very important one. God loves all persons. Be respectful of those we disagree with, even though we reject their beliefs about origins. We don't put down on people who think different. God loves them, and we need to love them and be friends. And I find this works if we really mean it. Our standard, our confidence is still the Bible. Our confidence is still faith in the Bible. But God is using science to encourage us. Thank you.
Well, thank you very much, Professor Brand. I'm sure you'll all agree that was an enlightening précis of a complex topic. Now, we've got opportunity for the next couple of minutes for some questions. So if I can get Dr. Standish and Professor Brand up again. And this is your opportunity to ask some questions. And
my question's for Professor Brand. I appreciated what you had to share. In terms of the mechanism of evolution being mutations, if that was something that was observed and that we saw on a regular basis, I could almost accept it. But my problem with evolutionary theory is not that the timescale's too long, but that it's far too short, in my opinion, because they say we only went from single to multi-cell organisms in the last 600 or so million years. So we're talking about 2 cells to a blue whale within such a short period of time. The sheer amount of mutations and added genetic material that has to happen and interconnected processes blows my mind. I'm just wondering, is there a problem also within the timescales? Because it just seems to be far too short a timescale, even if you could accept all of those processes as being credible.
I believe yes, it definitely is. The time that I would accept since the beginning, 6,000 to 10,000 years. Okay, and you get. Well, you look at the Cambrian explosion. You got all these organisms. All of a sudden, they're there. How did that happen? Actually, I was at a meeting of the Geological Society of America a few years back, where they had a discussion over this. There were some biologists who talked about what it would take to evolve all those different phyla of organisms that you find in the Cambrian. And the paleontologists who talked about this, they were trying to figure out how it could happen so fast. They knew there were no fossils down in the Precambrian below that, basically none. The biologists said, no, it needs 500 million years. So how do you bring these together? They're just 500 million years, but there's no fossils. Okay, so anyway, this is a conflict. And then you can look at other categories. For instance, most of the groups of mammals appear within a really short time up there here in the Cenozoic. It's an explosion of mammals. And again, bats appear suddenly, complete, and many other bats to whales. How do you do this? And there just really isn't enough time. So I agree completely with you.
We've actually just done a miracle. We've shut up a bunch of
Oh,
Australians.
not quite, not quite. I'm always the odd one. Could I read a little while ago John C. Sanford's book on genetic entropy, and I'm just wondering if you could just comment on the veracity of his work and whether it actually stands up. Because online, I don't have a scientific background, so online you read the counter arguments to it, and I'm just wondering from your perspective how that actually stands.
Okay, John Sanford is a very interesting fellow. He's a famous molecular biologist. He was a researcher at Cornell University most of his career. He invented the gene gun, if some of you will know what that is. But he started asking questions, and through a long process, he became a Bible-believing creationist. And he published this book, Genetic Entropy. And what he's showing is that it seems evident that our genome is degrading a certain amount. I've seen the figure of 1% per generation. I don't know if that's right or not, but anyway, it's degrading. And so if we've been evolving over 6 million years, and if our mutations are degrading our genome that rate, none of us should be alive. That's his basic point. There can't have been that much time for humans here.
Yeah, I know John as well. Here's the thing with these arguments. Nothing is a silver bullet. What you have to understand is every argument that is made in support of the biblical record of history actually does have a counter argument to it. You can think of these as being almost like chess pieces that you can move around. So you see predictable patterns in it all. The flip side is also true. Every argument that you see in favor or in support of the Darwinian view, the materialistic view, you are going to find that there is some kind of comeback to it. That it works in exactly the same way as question time in Parliament. You get one side jumping up and asking a very sneering question, and then the other side jumps up and sort of sneers right back at them. We've got to be kind of careful about those getting sucked into that sort of thing. Ultimately, with these questions, people who are thinking rationally will go with what really amounts to a preponderance of evidence while understanding that there will always be open questions. And so I am certain that there are comebacks to what John has proposed, and I'm certain that John can respond to those, and you could read his responses, and you can go back and forth and back and forth. However, when I look at the overall structure of the argument that he's making, it appears to me to be a reasonably strong one, while acknowledging, of course, there's going to be people out there on the internet who are trying everything they possibly can to shoot it down, because it is a strong argument.
Yeah, I agree completely. You can't get around critical thinking and analyzing these arguments. I'll make one other comment. An issue that does not have philosophical implications, you're likely to get better arguments on both sides. If it can't fit one philosophy, you're probably going to get poorer arguments coming from that side than you would expect.
I was nearly going to ask this question of Jacques Ducan before, but in his presentation he basically went back to what the hermeneutical models were that were being applied to Scripture in order to come up with the particular theories of how it developed and so on. And it seems to me, and so this is more of an observation than a question, but it seems to me that the presuppositions that or assumptions that one brings to the evidence is going to determine what your outcomes are going to be, your conclusions that you're going to draw. And given that postmodernism, for all its faults, has taught us that there's no such thing as a totally objective view of anything, wouldn't we be better off to acknowledge what our presuppositions are to start with when we're looking at the data?
Yes.
Yeah, I agree with that, yes, too. Absolutely. But let me suggest to you one thing that I found in reading, for instance, the anti-creation literature. We should each express our point of view to begin with. We have a distinct advantage in that which the other side does not understand. I read all this anti-creation literature. They obviously do not have a clue how an educated creationist thinks. But if we're going to be doing science, we have to understand how they think and how we think, how they interpret evidence and why, and how we would, and those of us who do that, we're all the time comparing these two and looking where we can test between them. And so we do have a distinct advantage if we think carefully.
One other question or observation, if you like. Recently I attended a conference on artificial intelligence where they drew a graph which told us that man-made intelligence will reach the level of self-replication and self-awareness somewhere between 2040 and 2050. And that's pretty incredible, and I can believe that it may not happen. However, we have in the cell, as been described already, and I won't use the term artificial, but an apparent intelligence that operates the systems. So what kicked it off?
Oh, okay. What kicked it off? Well, I can give you my answer. I mean, it's obvious. It's described in Genesis. Maybe a question related to that. Who was it? One of our gentlemen here talked about how you can have all these, I guess it was John Ashton, you can have all these components of a cell there, but it isn't doing anything until you can light the fire, so to speak, get all those processes going. And that's a serious problem for the theory of evolution.
I think that this also kind of relates to what you're sort of bringing up, and that is certainly I was taught to view things like cells in a very mechanistic kind of way. It's machines interacting with other machines. That's how, at least when I was in graduate school, we were taught about molecular biology. It's molecules and they're machines and they're all working together in that way. But I do believe that there is another productive way of viewing what's going on in cells and all of the systems that actually come together to allow something to be living, and that is from an information point of view. And this is not something I've come up with. There are plenty of other people thinking about it. In some ways, you can think about cells as information processing systems, where you have an incredible number of logic functions that are playing out in various different ways, as in the biochemical example that Ross was giving here earlier. You know, it's not just A becomes B becomes C becomes D, and there's a machine at each point. There are decisions that are made at each point. And so if you start thinking about these things in terms of information systems, that does kind of form more of a bridge between the idea of intelligence as well. But I don't think that this really relates to the question of artificial intelligence. I just hope that I'm around in 2040 to see whether they actually pull it off.
Well, it might be time for us to take a break. And thank you very much to our speakers again. Let's just thank our speakers. So we've got about a 20-minute break, and then it's time for the workshops. So does everybody know, and some of you won't know, where the white buildings are? Okay. I need somebody to help me. Yeah, they're over in the middle of the campus. Best way to explain that, somebody from Avondale.